So today we will be covering the topic genetic elements, sex types, conjugation and protoplast fusion. So genetics is the study of hereditary and the variation. So the arrangement of genes within organisms is its genotype, and the physical characteristics an organism based on its genotype and the interaction with its environment is its phenotype. Genetic information is contained in the nucleotide sequence of the DNA. So a gene is a nucleotide sequence that codes for a polypeptide, tRNA, which is the transfer RNA, or the rRNA, which is the ribosomal RNA. A particular organism may possess an alternate forms of some genes. So such alternate forms of genes are referred to as alleles. The cell's genome is stored in chromosomes, which are chains of double-stranded DNA. The genetic material of bacteria and the plasmid is the DNA. The two essential functions of genetic material are the replication and the expression. So now we'll come to the bacterial plasmids. So plasmids are an extra chromosomal genetic elements which are present in bacteria and rarely in eukaryotic organisms. In bacteria, these plasmids are circular double-stranded DNA molecules which contain the genes controlling a wide variety of functions. So plasmids are also a self-replicating elements, yet they are largely dependent on the host cell for their reproduction because they use the host cell replication machinery. So the first plasmid to be discovered was the sex factor or the F plasmid. This F here, it stands for fertility of E. coli K12. This plasmid confers the ability to an E. coli cell, that is the F plus, to conjugate with another lacking this plasmid. The F plasmid, they can exist in two alternative states, that is, it can either remain free in the cell or it can be integrated into the E. coli chromosome. So plasmids with such property are known as episomes. So now we'll come to the F plasmids. The F plasmid is a conjugative plasmid which is found in several bacteria, and the cell possessing the F factor is designated as F positive cell and acts as the donor. Then a cell which is devoid of this factor is designated as F negative or F minus cell, and it acts as a recipient during conjugation. So the cell possessing the F factor is the male, whereas the cell that do not possess or that do not contain the F factor is the female. So a part of the bacterial chromosome may also enter the recipient cell and it may lead to genetic recombination. Eventually, the F factor, it gets transmitted from the F positive cell into the recipient, that is the F negative cell. So such recipient, that is the F negative cells, therefore becomes the F positive cells. The F factor can exist independently of the host chromosome, or it may get integrated into it. So the bacterial cell in which the F factor is integrated into the chromosome is called the HFR, which means the high frequency recombination cell. The F factor integration, it causes a relatively very frequent transfer of the bacterial chromosome from the donor into the recipient cell, and this results in a high frequency of recombination. So now we'll come to the transposable elements. Under this, the DNA segments that carry the genes which are required for the transposition, that is, the movement of a piece of DNA around the genome and it consequently move out the chromosome, are termed as the transposable elements or the transposons. So these transposons, they behave like a lysogenic prophage, except that They originate in one chromosomal location and they can move to the different location in the same chromosome. So this transposable element, they differ from phage, that is the viruses, in lacking a virus life cycle and from plasmids in being unable to reproduce autonomously and to exist apart from the chromosome. So the simplest transposable element are the insertion sequences or the IS element. This IS elements was first discovered in the genes that controls the galactose utilization in E. coli. The IS elements are the normal constituents of the bacterial genome. So they may be present in the chromosome or as a plasmid. For example, these IS elements present in the F plasmid of E. coli, they are involved in the integration of the F plasmid with the bacterial chromosome, producing the HFR strains. So during the process, this genetic exchange between IS element of the plasmid and the chromosome, it takes place. So there are many IS elements in bacteria which differ in their nucleotide sequence. One important characteristic of all the IS element is the presence of 9 to 41 nucleotide long sequences at each end, that is repeated nearly identical but oriented in the opposite directions. So these sequences are called the inverted terminal repeats. In between the terminal repeats, a long stretch of DNA double helix, which contains several hundred to more than 1,000 base pairs, is also present. So now we'll see the transfer of genetic material. Sometimes, when two pieces of DNA come into contact with each other, some part of each of the DNA strand will be exchanged. And this is carried out by a process called crossing over, in which the DNA breaks and is attached on the other DNA strand, leading to the transfer of genes and possibly the formation of new genes. So genetic recombination is the transfer of DNA from one organism to the another. The transferred donor DNA may then be integrated into the recipient's nucleus by various mechanisms. In the case of homologous recombination, homologous DNA sequences having nearly the same nucleotide sequences are exchanged by means of breakage and the reunion of the paired DNA segments. So genetic information can be transferred from organism to organism through vertical transfer, that is, from a parent to a offspring, or through horizontal transfer methods such as conjugation, transformation, or transduction. So bacterial genes are usually transferred to members of the same species, but occasionally a transfer to other species can also be occurred. So now we'll see bacterial conjugation. The F plasmid, also called the fertility factor, it confers the donor characteristics, that is the sex pilus, to the bacterial cells. As mentioned above, the bacteria that have the F plasmid are termed as F positive or male, while those that do not have this F plasmid are called the F negative or female. Conjugative plasmids initiate gene transfer by altering the cell surface to allow contact between the plasmid containing donor cell, that is the F positive for male, and a plasmid lacking recipient, that is F negative or the female. Consequently, the sex pilus, it originates from the donor and it establishes a conjugate bridge, that is the temporary cytoplasmic bridge, that serves as the channel for DNA transfer from donor to the recipient bacterial cell. Further, the intercellular contact between the cells is established. So a copy of DNA from the donor cell, that is the F positive cell, is transferred to the recipient cell, that is the F negative cell. Finally, the complementary DNA strand is synthesized in both the donor cell and the recipient cell. As this recipient cell now contains this F plasmid, it behaves as a donor cell. If these F negative cells and the F positive cells are mixed in the culture, the entire population quickly becomes F positive cells. So plasmids, they may also integrate into the bacterial chromosome by a recombination event depending upon the extent of DNA homology between the two. After integration, both the plasmid and the chromosome, it replicates as a single unit. A plasmid that is capable of integrating into the chromosome is called an episome. If this F plasmid is integrated into the chromosome, it is called an HFR cell. So after integration, both chromosome and plasmid can be conjugally transferred to a recipient cell. Here, HFR cells are so called because they are able to transfer these chromosomal genes to the recipient cell with high frequency. In this process, the DNA is nicked at the origin of transfer and is replicated. Further, one DNA strand begins to pass through a cytoplasmic bridge to the F negative cell, where its complementary strand is synthesized. Along with the portion of integrated plasmid, the chromosome is also transmitted to the F negative cells. So the bacterial connection usually breaks before the transfer of the entire chromosome is completed. So the remainder of the F positive plasmid rarely enters the recipient. So usually only a part of the HFR chromosome as well as the plasmid is transferred during conjugation, and the recipient cell, it does not receive complete F factor. After conjugation, the HFR cell remains HFR, but the F negative cell does not become F positive and it continues to remain as F negative. However, the transferred chromosome fragment recombines with the chromosome of F negative cell, thereby transferring some new property to the recipient cell. So now we'll see the protoplast fusion. So protoplasts are the cells of which the cell walls are removed and the cytoplasmic membrane is the outermost layer in such cells. So protoplast fusion is a physical phenomenon. During fusion, that is during protoplast fusion, two or more protoplasts, it comes in contact and adhere with one another either spontaneously or in the presence of fusion-inducing agents. So protoplast fusion is an important tool in strain improvement for carrying out the genetic recombination and developing hybrid strains. Protoplast fusions are used to combine genes from different organisms to create the strains with the desired properties. So let's see the methods of protoplast fusion. Protoplast fusion can be broadly classified into two categories. The first one is the spontaneous fusion and the second one is the induced fusion. So let's see the spontaneous fusion first. Protoplasts during isolation often fuse spontaneously and this phenomenon is called the spontaneous fusion. During the enzyme treatment, protoplasts from the adjacent cell, it fuse through their plasmodesmata to form the multinucleate protoplast. While in the case of induced fusion, it is the fusion of freely isolated protoplasts from different sources with the help of fusion-inducing chemical agents. Normally, isolated protoplasts do not fuse with each other because the surface of the isolated protoplast, it carries the negative charge around the outside of the plasma membrane, and thus there is a strong tendency in the protoplast to repel each other due to their same charge. So this type of fusion, it needs a fusion inducing chemicals which actually reduce the electronegativity of the isolated protoplast and allow them to fuse with each other. The isolated protoplast can be induced to fuse by two ways. The first one is the mechanical fusion and the second one is chemofusion. First, we'll see this mechanical fusion. In this process, the isolated protoplasts, they are brought into intimate physical contact mechanically under the microscope using a micromanipulator or perfusion micropipette. Then, in the case of chemofusion, in this method, chemicals are used to induce the protoplast fusion. So, chemicals such as sodium nitrate, polyethylene glycol, etc. are employed for this method. Chemical fusogens cause the isolated protoplasts to adhere each other and it leads to tight agglutination followed by diffusion of the protoplast. This chemofusion is a non-specific, inexpensive and can cause a massive fusion product. It can also be cytotoxic and non-selective and having less fusion frequency. So at the end, I would like to add that plasmids are an extra chromosomal genetic materials or genetic elements which are present in bacteria and rarely in eukaryotic organisms. These are not essential for the survival of the bacterium, but they confer certain extra advantages to the cell. And these plasmids, they multiply independently of the chromosome and are inherited regularly by the daughter cells. Then talking about their application, they are used in genetic engineering as a vectors and also for genotyping method, plasmid profiling is a very useful one.